Okay, I think, yeah, it is recording. Okay, so today we're going to be talking about chapter 20. And chapter 20 is going to be talking about blood vessels. And if you have looked at it already, you know that like really second half of this chapter is all the naming of the blood vessels. And that's why I chose this image right here, memorize all the blood vessels. So a good thing is if you remember your bones, then this should be a little easier to memorize these. So you'll see some similarities between certain things from A&P1 in this chapter whenever you're memorizing those blood vessels. So if you haven't started yet, start on it today. Try to at least look at one diagram and try to get it down, and then just add more and more diagrams later. There are three main blood vessel types. We have arteries, veins, and capillaries. Arteries are blood vessels which carry blood away from the heart, and veins are going to carry blood towards the heart, and capillaries are going to serve as sites for exchange of material. While the capillaries are the smallest vessels, only consist of single layer of thin endothelium, our arteries and veins are going to contain layers known as tunics. So we'll be talking about these layers, and we're gonna refer to them as tunics. These tunics are tunica intima or interna. So this is going to be the internal layer. Then in the middle, you'll have tunica media, so media for middle. And then tunica externa, that should be pretty obvious that externa would be external thing. The internal space where the blood is located in a blood vessel is called lumen. So now I want you to look at the blood vessel hold on, and list some differences you observe between artery and vein. And Malia, you say you can't hear me because my voice is cutting in and out. I don't know if that's my connection or if that is your connection. Okay. So it might be your connection. I'm sorry, but I'll post the recording, and that should not be cutting in and out because I'm recording to my computer and not to cloud. So it should have everything smooth whenever I post the recording. Okay, so look at this, and you should be able to click on the picture, and I didn't, or maybe you're not, but just looking at that image that you should be seeing right now if you are on your device. Let me actually share my iPad here with you. Looking at that picture of artery and vein next to each other, what are some differences that you see here? And okay, let's do this. Okay, so now I'm sharing this. All right, good. What are some structural differences you are seeing? Like, just looking at it, and I don't know. Yeah, you can actually click on that picture and it will become bigger. So what are some, like, just by look, do you see anything that's different about these? Okay, we got thickness. Okay, good. Anything else you notice? Okay, good. Valves is a good one. Yeah. Okay. The tunica media is wider on one. Specifically, it would be on your artery, right? So arteries have valves. That's a good one. And I mean, wait a minute. Victoria, did you mean that? No, reading that. Okay, pressure is higher in the arteries. That is correct. Not something you can actually really see from just looking at the picture, but if you're thinking about it, the thickness of the wall actually is going to have to do with the fact that in the arteries, there's going to be higher pressure, so they need to be thicker. With the valves, it's actually the opposite. Arteries do not have valves. Veins are the ones that have valves. And we'll see later on that this has to do with the fact how the blood is traveling through these vessels. And arteries really don't need valves because it's the blood that is, it's the heart that's propelling the blood through. But veins are already kind of removed from the effect of the heart. And so they're going to rely on other mechanisms to move the blood through them. So just to kind of summarize what some of the differences are that you can see, and there's a table for this where you can see all of the differences there. But some things that just visually you can observe are that arteries are thicker. So they have those thicker walls. They have smaller lumen than veins. For this reason, veins tend to actually collapse when they're not filled with blood. So because the veins are so thin, or so much thinner than arteries, if they don't hold blood in them, they will collapse. And arteries generally are going to hold their shape. And this is something what will help you identify them on the microscope slide. You'll see that artery is going to usually have a nice thick round shape, but the vein is going to be much thinner and kind of collapsed around it, or right near the artery. The veins have valves. These are going to prevent backflow of blood through them. Arteries carry blood away from the heart. We already said that. And but veins, they are going to have, they're going to rely on skeletal muscle, surrounding skeletal muscle pump or respiratory pump to push blood through them. And that's why they have to have those valves. Veins don't have elastic lamina. And this is an unlabeled picture that I shared with you here on that collaboration board. But veins actually don't have elastic lamina. They are elastic. They have elastic fibers. They're just not concentrated in elastic lamina like what we see in the artery. For some of the other differences between arteries and veins, look at that table 20.1. And that's going to kind of solidify everything, all the differences for you all together. Okay, so let's go on here. And why am I not hiding the names? Hide there. Okay, so from the inside out, the 3 tunics of muscular artery are tunica something, something, something. So 3 tunics, how does it go from inside out? So start with the internal one and end with the external one. You should be able to drag those boxes, I think. No, I don't know, because now I have this on my screen twice. Okay, let's see what happens if I click done. Oh, wait, what? I'm running it from my computer. Okay, close. What happens if I do this? Okay, y'all got it. Okay, it was weird because when I hid the names, I wasn't seeing if you guys were answering. So, okay. That's that. So let's go on and look at the tunics here. So here's that same picture that we looked at before, but now it is labeled. And so we see those tunics and their sublayers. So these tunics are going to have some sublayers here. So tunica intima, the internal layer, is composed of your simple squamous epithelium. This is going to be on the lumen side, so it's the innermost layer. And then basement membrane as we're going towards the outside of the vessel. Okay, so you have endothelium, you have basement membrane on the other side of that endothelium. Then tunica media is going to be composed of smooth muscle and elastic fibers. The tunica media is responsible for vasoconstriction because it has that smooth muscle in it. And this layer is going to respond to various stimuli. We'll be talking about what these stimuli are. It will either vasoconstrict, which means it will get smaller, okay? It will constrict, or dilate, like it will open up. So vasodilation is when the vessels open up and they increase their lumen size. And the outermost layer is the tunica externa. And the tunica externa is composed of areolar connective tissue, and this is also going to contain many collagen fibers and elastic fibers. This layer also houses vasa vasorum, which are vessels of the vessel, and these are going to supply your larger blood vessels that need nutrients and oxygen. So if you remember how heart needed to have its own blood supply, remember that circulation, the coronary circulation of the heart, it was bringing blood to the heart itself. Even though heart had blood going through it, that blood that was going through the heart really wasn't supplying the heart muscle with anything. It was just passing blood through. So heart had to have its own circulation. So large blood vessels also need to have their own circulation, because the blood that is going through them, through the lumen, it's just passing through. And let me go ahead and check the chat here. Okay. All right. Okay. I was just catching up on the chat here, and let me copy the code here for you. And there it is, the code again, because I know that chat doesn't update when you, like, if it kicks you out and you come back or something like that. So now it's in the chat again, and here we go. So anyway, so those are our tunics in kind of in general. Notice that you have some sublayers here. The artery that you're seeing here, that's labeled here, this is actually a type of a muscular artery. So you're going to see some layers that I haven't mentioned in the general description of the tunics, but we will describe them as we talk about the different types of the blood vessels or of the arteries. Okay. And again, we've pointed out some differences here that are kind of obvious there. And you can see those elastic layers here in that artery, and you don't see that elastic layer in the vein. All right. So let's see if you are able. Yeah, I tried to draw like boxes here. I wasn't using the computer. I was just using my hand here. So see if you would be able to label the parts of the artery. And I'll have this is just for your practice. Notice that I grouped some of the boxes together. So the easiest probably would be here for you to create a text box for the individual thing. Okay. So if you click this thing, the double T there, that will allow you to enter a text box and put that in there somewhere. So you would say endothelium, I'll give you that one. And then you can, whoa, boom. See, kind of like that. And it doesn't have to be super pretty as long as it's in the right space. So while you're working on this, I wanted to point out something. So I created a link on Blackboard that is to a Google Drive. In order to access this Google Drive, you will have to sign into your browser with your MyTCC credentials. Once you sign in, it should give you access to unlabeled images so that you can practice this labeling. It should also give you access to like my super old PowerPoint notes that I'm no longer using, but I used to use, and it's kind of just kind of my outline. There are some things that needed to be changed that I haven't changed. Like there are some typos and things, so they're old, okay? But I thought maybe they would be helpful to you. Kind of as an outline, so you can get a look at that. And also the histology file is in that Google Drive as well, so you can find it there or on the SLC webpage. All right, I see like a couple people are almost done. Okay. If you're looking in the textbook at this image, you will notice that the basement membrane is labeled as subendothelial layer, I think, or subendothelial membrane. I don't know what they're calling it, but it is the same as basement membrane. And it's kind of easier for me to say basement membrane, but they're used interchangeably. So just FYI, subendothelial layer and basement membrane are going to be the same. So let me see. Okay. All right, I have one here that I wanted to go ahead and, how do I share this? Oh, I can't share it because it's in progress. So if you're done, hit submit. I know it's kind of tricky to label that, so. All right, I think I can share this now. Yes, so once it's submitted, I'm gonna go ahead and share this one because that's the only one that has been completed. And I wanna say, and it should not say who it is. I should have the names hidden, but so if you're looking at this, you have the endothelium, then you have subendothelial layer, which is the same as the basement membrane, and then internal elastic lamina. Good job. That it together makes up tunica intima. Okay, so tunica intima is those three layers. And then when we go on to the tunica media, where tunica media should be after that, where the two lines come together into that box. The first one, so you have external elastic lamina. The other thing that makes up the tunica media should be the smooth muscle. So make that correction for you. That is your smooth muscle there. So it's smooth muscle and external elastic lamina together make tunica media. And then the bottom box, it is tunica externa actually, but it does contain vasa vasorum. So again, that was close. Vasa vasorum would be actually those little like blue and red dots that you see within that layer. And again, that's the vessels of the vessel. So I'm going to unshare that, but good job. And we have another one here. Well, that one wasn't all the way completed, but I think most of it was because you're kind of trying to figure out how to put in those boxes. So if you're on your phone logging into this or on a tablet, you can use your finger and write it in, or just keep in mind that you should be able to label these come the exam time, okay? So now to kind of go to the other side of this, because we labeled the artery, okay? And that artery was muscular artery, just FYI. Now we're gonna look at the vein. Vein is a little different. It is missing those elastic laminae. But the other things are going to be the same, kind of in the same order, minus the elastic layers, okay? So and it's fewer boxes for that reason, okay? So what do we have here? And actually, I think I can just write with my pencil here. Okay, Okay, so some of you are pulling this up. And yeah, whenever you're looking at this on your own, if you are looking at this on your own later on, you could screenshot this, print it out, label it for just for practice. Okay. All right, let me see. Okay, good. Yeah, one of those boxes is not a layer, it's a structure inside of the vessel, inside of the vein. Good. And yeah, if you're on your phone, it's kind of difficult to write with your finger. Good. All right, good. So I'm watching your submissions there. And yeah, good. A couple of you are already done. Oops. All right, let me see. Oh yeah, that's fine. Yeah, sometimes that space is not, and I know that those boxes are probably not all that huge. Okay, that's perfect. So I'm gonna go ahead and share this one. So everybody should have this on their screen right now. And so here you see that on the vein, we have endothelium and the subendothelial layer, and those two together create a tunica intima. Now there's not that elastic layer here, right? Then we have the valves here. The tunica media in a vein is just a smooth muscle. So smooth muscle and tunica intima in the vein are the same thing. In the artery, tunica media is the smooth muscle and the external elastic lamina, okay? But in the vein, tunica media and smooth muscle are the same thing. And then the outermost layer is the tunica externa. So great job if you got this. And so let's go ahead and keep going. So let's talk about those types of blood vessels, because I have been talking about the muscular artery, right? So on here, we can see there are different types of arteries and veins. So we can differentiate them, and it's basically going to be based on their size, but also the layers are going to be quite different, or not quite, but a little bit different there. So the different types of arteries actually are going to have specific names. The largest arteries are your elastic arteries, and these are going to include the aorta and the pulmonary trunk, and then the branches of aorta and pulmonary trunk. So the first vessels that come off of these are also elastic arteries. So number wise, we don't have too many of these elastic arteries. Keep that in mind, because we're going to come back to this concept of, you know, like numbers versus the size of the vessel. All right. These arteries have elastic fibers. They're dispersed all throughout the tunics, and therefore they do not have elastic lamina layers. So elastic arteries do not have internal or external elastic lamina. Okay. A lot of times people want to say, oh, the ones with the elastic lamina are elastic. No. Okay. Elastic arteries are called elastic because all of their layers have elastic fibers. Okay. So they do not have internal or external elastic lamina layers. Elastic arteries do not have internal or external elastic lamina. Okay, remember that. These arteries are known as your conducting arteries. They're going to bring blood towards a specific area, general area. The type of artery that we labeled on our on that previous the previous picture where we labeled the artery, that is your muscular artery. These are still pretty stretchy, but they're less stretchy than elastic arteries. They have thicker muscularis layer. That's why they're called muscular arteries. And they also have those internal and external elastic lamina layers. So their elastic fibers are concentrated in those two distinct layers. The elastic fibers in the muscular artery do not go through all of the layers like we saw in the elastic artery. So these are known as distributing arteries. They take blood towards a specific organ usually. These arteries are going to branch into smaller arterioles, and the arterioles can vary quite a bit in size. These are smaller, that's what the muscular arteries branch into, and they can vary greatly in size. Some of the largest arterioles are going to have all three tunics, but the smallest arterioles, they are going to have just the endothelium, basement membrane, and then a single layer of the smooth muscles. So, and you'll see that actually when we look at the capillary exchange picture, you'll see that single layer of smooth muscle. The blood from these arterioles will flow through capillary bed then. And then as it passes through capillary bed, it will be picked up by venules. Those are your smallest veins. These venules will merge into small to medium veins, and then ultimately into the largest veins. And the largest of the veins are the vena cava, the superior and inferior vena cava. So that's kind of how that goes. And yeah, so veins don't have any specific names, so we just venules, and then small to medium veins, large veins, and then, well, the vena cava, I guess, have their names. So those are our types of vessels. Let's go, our types of the arteries and veins. Let's talk about capillaries here. There are about 10 billion capillaries in a human body, and this makes up about 60,000 miles of capillaries. And if you like metric system like me, it's about 100,000 kilometers of capillaries. There are three different kinds of capillaries: continuous capillaries, which are completely lined with endothelium and surrounded by a complete basement membrane. This is most of your capillaries in your body. So majority of your capillaries in your body are continuous capillaries. The second type are fenestrated capillaries. And as you can see on the picture, there are some holes on the endothelial cells, and these are known as fenestrations. These are very, very thin areas of the cell membrane. They're really serving as pores, and these allow larger particles to come through, to go from blood into the outside of the vessel. Like sometimes some small proteins, things like that, but not formed elements. Okay, they also allow for a lot of fluid to exit out. And so we're going to see these in areas where a lot of traffic or a lot of exchange has to happen, such as in kidneys, when we're filtering fluid out and trying to produce urine ultimately. Also in small intestine, where you're absorbing all the nutrients from the digestive system, all the fluid and nutrients from the digestive system. And also most endocrine glands are going to be fenestrated capillaries. And the last type of capillaries are sinusoids. And these you will see in the next unit when we talk about spleen. You will see sinusoid, and it's not actually even going to really look like a capillary. It's kind of like a hybrid between a capillary and a space. That's why it's not called a sinus, but it's not also called like really true capillary. It's a sinusoid. It's kind of something in between. These are going to have large gaps between the cells that make it up. And also they're going to have either incomplete or absent basement membrane. So look at that image and you can see that there are large spaces, and these spaces are so large that they allow for formed elements to move through. So we would see these in places where we have to move large particles in and out of blood, such as formed elements. So For example, red bone marrow. This is a site of hemopoiesis. This is formation of formed elements. So once your bone marrow makes these formed elements, they have to enter the blood somehow, okay? And it would be in the form of sinusoids. So these new formed elements enter the blood into this or through the sinusoid. Also places where we have to recycle formed elements, such as liver and spleen, are going to be full of sinusoids so that these formed elements can exit the blood when they get worn out, so we can recycle them. So those are our types of capillaries. Now, capillaries don't actually generally function as a single capillary. They are found in groups that we call capillary beds. And so capillary beds are collections of several capillaries. Let's look at it. Oh, wait. I did not include my picture of a capillary bed, but if you are looking at your textbook, you should be able to see the capillary bed image. And you have an arterial. I cannot believe I forgot that to include that picture. Make a note to myself so that whenever you're looking at this on your own, you will have that image and you will know what I'm talking about. But in the capillary bed picture, you would see an arteriole, then you would see a venule, and in between you see a network of capillaries. Actually, let me see if I can share a picture, because I have that picture somewhere here. And yeah, right here, I can share this, I believe, with you so that we are looking at it. I don't know how it escaped me. Okay, here's that image right here. And so hopefully you're seeing that as it's being shared. But you have that arterial end, okay, you have that arteriole. And here is where you can see that single layer of smooth muscle, those little pink cells that are wrapped around that arteriole. That's a single layer of smooth muscle. And then there was going to be a branch off of that arteriole that we're going to call medarteriole. And this one is going to lead it's going to be like an origin of your true capillaries. The distal end of this, kind of the central vessel that you're looking at there, is called the through-fare channel. And then off of the medarteriole, you see branching the true capillaries. At the origin of the true capillaries, you see those precapillary sphincters. These are smooth muscle cells that are able to contract or relax. So these smooth muscle cells are going to regulate passage of blood into those true capillaries. So when they constrict, the blood is actually not going to get into the true capillaries. It's just going to flow all the way through to through-fare channel and then into the venule. If they're open, then the blood can go through those true capillaries. And most of the exchange of materials is going to happen in the true capillaries, okay? And we regulate the passage of the blood through capillaries, and there's a reason for this. So remember, we have 10 billion capillaries, that's 60,000 miles of capillaries. Well, our blood volume is only on average about 5 liters. So it's actually given that at any given point in time, if you were to stop blood flow and just look at where the blood is at any point, you would see that only about 5% of capillary beds are filled with blood. The rest of them are not. The rest of them are in a closed state, as you see on that bottom part of this image. And this is because we simply don't have enough blood to fill all of the capillaries at the same time. Now, these capillary beds take turns, okay? They alternate all the time. And these precapillary sphincters, they contract rhythmically about 5 to 10 times each minute. So it's not like you're keeping, under normal circumstances, you're not keeping capillary bed closed for too long. And yeah, anyway, so these capillary beds do take turns. So that's our capillary bed. Let's see where I want to go next. So that's that image. All right. Let's go back to my iPad. And I always have to run this way. Okay, so let's go to the next slide here and let's see if we remember what we were doing. So on your screen, where you pulled up the Nearpod, play a little game and see how much you remember. Okay, I'm gonna. Okay, I'm hiding your names. So we don't see who's who right now. I'm gonna go ahead and get started. And I'm not answering, I'm not gonna give you the answers. So all right, good job. So I was the one with the 0 points because I didn't play. So anyway, let's see. Let's see. Good job, Tim, Leah, Victoria. Good job. So anyway, that was just a little review. It's okay if you didn't score all that great. You still have time. All right, let's look at distribution of blood. So where is blood found in our body? And you can see this on this diagram here, and this is at rest, okay? It's going to change when we are going through physical activity, but at rest, about 70% of the blood in your body is in the systemic circulation, but 18% is in the pulmonary circulation, and then the remaining 12% is in the heart. Majority of your blood is going to be in your systemic veins, and this makes your veins serve as blood reservoirs. During physical activity, then blood can be redirected from these blood reservoirs into active circulation. But when you're at rest, it's just there. And this is because, remember, veins have a larger lumen, so they're able to accommodate more blood than the arteries. And yeah, we don't have to have all of our blood volume in the active circulation at rest, but then during physical activity, there's going to be vasoconstriction. This blood is going to be, your veins are going to constrict, and this blood is going to be added into your active circulation so that you can move more things through your body as the body needs it. Now we're going to talk about the blood pathways. And so we can see that on this image right here. I know it's kind of small on this, but in your textbook, it's a little larger. And I'm so glad we're seeing the sun, but now it's like coming my way again. It feels like forever since we saw the sun, right? So the blood pathways, we can describe them as either simple pathway or alternative pathway. Simple pathway is simply where you have a single major artery that supplies a tissue or an organ, and then there is a single major vein that leads out of that organ. A perfect example of this is spleen, where the splenic artery would be supplying the spleen, and then the splenic vein, just a single vein, is getting the blood out of the spleen. A lot of our tissues and organs, however, rely on alternative pathways. And alternative pathways are going to include anastomoses and the portal system. So anastomosis, just FYI, anastomosis, SIS is a singular, SES is a plural version of this word. So anastomosis versus anastomoses, the SES. So we can talk about arterial anastomosis. This is where you have several arteries supplying the same organ or region. An example of this would be your superior and inferior epigastric arteries or circle of Willis in your brain. So when you start looking at these vessels, at these diagrams, you will see what we're talking about and how they are interconnected. There are also anastomoses between your coronary arteries, but these are so small, so narrow that they actually don't serve as functional connections, meaning that blood flow, if it needed to be redirected, it wouldn't be sufficient to keep the heart alive. So for that reason, we consider coronary arteries functional end arteries. So maybe you remember that from chapter 19, maybe coronary circulation. So that's that. The venous anastomoses are more common actually than arterial anastomoses. And this is where several veins are going to drain a certain region or an organ. So for example, basilic, brachial, and cephalic veins, these are going to drain the upper limb. And that's an example of venous anastomoses. Again, when you look at that diagram, think back to this concept and see what that means in an actual example. Because this diagram has it, but you can, when you actually look at that arm, you will see how they are connected with each other. There are also dorsal venous arch of hand and foot. That's a great example of venous anastomoses as well. And then the last, one other thing that I forgot or almost skipped is arteriovenous anastomosis. And this is actually a shunt. So this is where we direct blood from an artery and we do not go through the area. We don't go through that capillary bed. We skip it. We bypass it. It's a bypass. It's a shunt. Okay, so arteriovenous anastomosis is where you connect artery to a vein and you skip the capillary bed or organ or something. The portal system is where you have two capillary beds connected to each other by a portal vein. So you have blood directed or delivered to an area. That one is going to spread through capillaries. It's going to do its thing, go through exchange, perhaps pick up something. Then that blood from that area will drain into a portal vein, and that portal vein goes somewhere else where it branches again into capillaries. And then the blood goes through some other things, depending on what portal system we're talking about. And then it is returned into the circulation. So I have a question for you. And let's see if you know the answer. Okay. So what is the name of one portal system we already talked about? And I want you to think back to endocrine system chapter. So we've already talked about a portal system. There will be another portal system we'll talk about later. So I'll give you like a minute. So if you want to, you can go back and look it up. Ooh, somebody gave me both. Well, two important ones. All right, 10 more seconds, 7, 5. Good. All right. So the answer is the one that we've already seen was the hypothalamo-hypophyseal portal system. And this is the portal system that passes the releasing hormones from the hypothalamus to the anterior pituitary. And it was, I mean, on a scale like size-wise, it was small. It is very important, but it was small. There is another portal system that we will be talking about when we talk about digestive system. And this is the well, actually, you will see it in the vessels here, but we'll kind of talk a little more about it in digestive system, and that's the hepatic portal system. And this is where all the capillaries from your digestive system that are absorbing all the nutrients from your GI tract, they're going to merge together into the hepatic portal vein, and then all this blood from the digestive system or from from GI tract is going to be directed to liver first. We're not adding this blood to the main circulation yet. The liver is going to receive this blood, it's going to process it, it's going to metabolize various things before we allow this blood to return or to enter the main circulation. Okay, so good job with that. And let's talk about this. Okay, the cross-sectional, and this is right before we're going to be talking about the capillary exchange here, and that will be the last thing that we'll talk about today. But before we get into that, let's talk about the cross-sectional area versus velocity of blood flow. So, and really I should have added the total cross-sectional area to this, which is in your picture here. So this graph is showing you the relationship between the total cross-sectional area of blood vessel type and the velocity or the speed of blood in that vessel type. So you can see that the relationship is inverse here. The smaller the total cross-sectional area is, the faster the blood flows through that vessel. It is really important to note, though, that we're talking about the total cross-sectional area here and not a cross-sectional area of an individual vessel. Okay. What that means is, okay, let's look at the elastic arteries, for example. Okay, remember me saying that we only have a few elastic arteries? We don't have that many of them. Individually, they're large, right? But you only have a few of them. So if you add the total cross-sectional area of those few vessels, the total cross-sectional area is not going to be that big, okay? But if you look at capillaries, okay, we've got 10 billion of them. Each capillary is super tiny. It's about the width of an erythrocyte, okay? But if you add all 10 billion of them, your total cross-sectional area of all these vessels combined is going to be much, much larger than those few larger vessels that you have, okay? So total cross-sectional area refers to all of those vessels of that type, all right? So you can see from this picture that your capillaries have the largest total cross-sectional area. Your elastic arteries and your largest veins have the smaller total cross-sectional area for their type, and the blood flow is going to be inversely related to that. So what that means is in the large vessels, the blood goes fast. The smaller the vessels get, the blood goes slower, okay? The velocity of blood flow, okay, where was I gonna go with this? So the speed or the velocity of the blood flow has to do with blood pressure also. The faster the blood goes, the higher the pressure is in there. So I want you to think about a water hose, okay? When you turn on the water, if you just turn it a little bit, the speed of that water is not that high, and it's also not coming out with a great pressure. If you open it up more, if you add more water into that water hose, if you allow more water to go through, it's going to go faster and it's going to go with a higher pressure. That's kind of how it works with our blood as well, okay? So you don't want to have the same velocity of blood and the same pressure in your capillaries as you have in your aorta. Aorta is very stretchy, it's very thick, and it receives blood as the heart contracts and it squeezes the blood into that aorta. You can't do that to a little tiny capillary. It would bust, right? It would burst, it would rip. So it's important to have the velocity lower in those smaller vessels. So that's what you're going to be seeing actually, is that, and we call this blood pressure gradient. Okay, you're going to see that the blood is going to slow down and the blood pressure is also going to decrease as you get into smaller and smaller vessels. And that's that. This is important so that you're not damaging those smaller vessels. And it's also important so that when it's at the capillary level, you have enough time to absorb things from the blood that you need to. If the blood moves too fast, you're not going to be able to absorb all the stuff that you need to absorb. So, slowing down in those smaller vessels is important. So that's our total cross-sectional area versus velocity here. Now, I do want to point out one thing on this graph that you might notice, and that is that even though in the vena cava, you have the total cross-sectional area about at the same level as with the elastic arteries, notice that the velocity is never going to reach the speed as we saw in the elastic arteries. And this is because after the blood enters the capillaries, it just continues to slow down and the pressure just continues to decrease. At this point, I mean, yeah, your heart is the main pump that pushes the blood through. But by the time we get to capillaries, the blood is no longer pulsatile. The pressure is much lower. And in the veins that now have larger lumen, the pressure in the veins is not going to be nearly as high as it's in the arteries. Veins are going to rely on the skeletal muscle pump. So skeletal muscle pushing on the veins from the sides and literally squeezing the blood inside so that it moves. Or a respiratory pump, which basically works the same way, but it involves the changes in pressure in your thoracic cavity and your abdominal cavity. And that's never going to be as strong as the force of the heart as the heart squeezes the blood into the vessel. And so that's why the velocity here is never going to be as high as in the arteries. So that's my last slide on here for now. What I'm going to share right now is something else. Where is it? And that's here. Actually, I could have just kept it there because I still want to share the iPad. So we're going to talk about capillary exchange now and some pressures that are associated with capillary exchange here. Okay, so what I drew in here, and you might follow along with the image for the capillary exchange in your textbook, which is very busy and it has a lot of numbers. So I want to simplify this a bit. Okay, so here's a simplified version of a capillary. Okay, you have the artery or arterial. So this is arterial, and then this is our venule here. All right. And the blood is going to go in this way. Okay. And when it is here, as it enters this, the first part of the capillary, right here, it's about 40 millimeters of mercury. mmHg, it's a unit of pressure. So this is 40 millimeters of mercury. There are several things that are responsible for things moving in and out of the blood. Okay. So as the blood is entering the capillary, there is pressure that the blood is putting onto the capillary from the inside. Okay, we call this pressure blood hydrostatic pressure, and it pushes on the walls of the capillary and it allows for fluid to be pushed out of the capillary through those intercellular clefts. Okay, that's how it's coming out. Remember those intercellular clefts? They're associated with capillaries. Okay, so we're gonna see, I like really needed a thicker thing. All right, so this is hydrostatic pressure of blood. As this blood pushes on the capillary walls, okay, on the walls of the capillary, fluid is going to be pushed through those intercellular clefts into the interstitial space, which is like right here, okay? And this is B. It doesn't look much like a B. Okay, so that's hydrostatic pressure of blood. And when I say fluid is being pushed out of the blood, what I mean by this is it's the plasma, blood plasma without the proteins. Okay. There is also, and I said without the proteins. So you know that blood contains in the plasma, it has plasma proteins. Okay. There's these plasma proteins. That's going to be these blue dots. Okay. They're everywhere, but we're not going to be pushing them out into the interstitial space. So they remain in blood, and that's why I'm trying to make them kind of evenly spaced out throughout. Okay, these are mostly your plasma, most of your plasma proteins are transport proteins. So they really don't need to exit the blood. They need to remain in the blood because they're transporting things through the blood. Okay, so the plasma proteins remain in the blood. There are only very few proteins that will not be transport proteins really that are in the interstitial space here. Okay, the presence of these plasma proteins inside of the vessel, inside of the capillary, and the absence of most proteins in the interstitial space creates a concentration gradient. Okay, these proteins that are found inside of the capillary are going to exert what we call colloid osmotic pressure. They're going to attract water. They're going to pull water towards them. So there's this pull of water or fluid from the interstitial space into the capillary. Okay, so this is kind of pull of the water inside. Okay, and this is colloid osmotic pressure. When you hear plasma proteins, I want you to think about colloid. When you hear colloid osmotic pressure, when you hear the word colloid, you think proteins, okay? And proteins pull water towards them, all right? So there's this kind of almost like a tug of war kind of thing where the hydrostatic pressure is pushing fluid out. The proteins that are inside of the capillary are trying to pull the fluid in, okay? So now, who's gonna win? At this end, where we are looking at right now, right here, okay, your hydrostatic pressure is larger than the colloid osmotic pressure. And for that reason, you will see a net movement of fluid out of the blood, okay? And this is known as filtration. Okay, so this is where the fluid from the blood, which now contains nutrients and all kinds of dissolved substances that are in the blood that need to go to the cells. This is how this fluid gets into that interstitial space. And this is what bulk flow is really, is what most of the substances move in this bulk flow. In this filtration process. So what's happening as you're filtering fluid, as you're going along this vessel this way, you're losing all this fluid in the filtration to the interstitial space. So the volume of the blood as you're moving from the arterial end towards the venous end decreases. Since the volume decreases, your pressure decreases. You have less blood, less and less blood as you're moving towards the venous end. And for that reason, what's happening is your hydrostatic pressure of blood is no longer as large at the venous end. Okay. I should have probably made that a little smaller. Okay. I have trouble with making these arrows exactly how I want them. I always make them kind of messed up. But anyway, so then you have this colloid osmotic pressure that really is not changing, and I can never make that arrow the same. But the COP, okay, maybe make this one a little bigger so they look the same. Maybe. Okay, the colloid osmotic pressure is not changing. It remains the same. And the decreased hydrostatic pressure is now lower than the colloid osmotic pressure. So now the colloid osmotic pressure, the pull of the proteins is stronger than the push of the blood. And so what we're going to see here is the opposite. We're gonna see the blood returning, or not the blood, the fluid returning back into the blood at the thoroughfare channel area, okay, at the venous end. So this is called reabsorption. Okay, so that's reabsorption, and it really goes there. Okay, it doesn't go into the venule, it goes back into the capillary. Okay, and so basically you're running this fluid full of nutrients and full of these substances for the cells through the area. Cells, which right here would be cells. Okay, I didn't draw them. Okay, but you'd see cells there. Cells are getting all these nutrients and all that. And at the same time as this fluid flows through, it's picking up carbon dioxide, it's picking up all the metabolic waste from the cells and all that, and it's returning back into the blood, and we end up with our deoxygenated blood full of, taken back or taking the waste away from the area, okay? And this is what describes bulk flow. Now, one thing that I haven't mentioned is that there is a part of the blood that actually, part of the fluid that doesn't get reabsorbed. So not all of the fluid that flows through gets reabsorbed back into the blood. There is some fluid that will be taken away by special structures. Okay, this is lymphatic capillary. And we're going to find lymphatic capillaries in most vascularized tissue. Okay, so where you have blood capillaries, most likely you're going to see a lymphatic capillary there. Well, there will be some exceptions that we will talk about when we talk about lymphatic system, but in general, most places where you see a blood capillary, you're going to have a lymphatic capillary. The lymphatic capillary is going to absorb anywhere between 10 to 15% of the fluid that is filtered out. So lymphatic capillary will take this. This is to monitor that area for any sign of infection that could be happening in that area, okay? So, and that will kind of, again, come together a little nicer when we actually talk about inflammation, and you'll see how this monitoring works. Okay, so some of this fluid is going to be reabsorbed by this lymphatic system. So not all of the fluid is returning back at the venous end. And what you're going to end up here with, as far as pressure goes, at the venous end here, is about 20 millimeters of mercury. So our blood pressure is going to decrease steadily as we start in the aorta. By the time we get to the capillaries, it's about 40 millimeters of mercury. It's not even pulsatile anymore. Okay, so notice that it's a single number that I'm giving you, but you know that when you go to the doctor and they measure your blood pressure, they give you two values, and that's the systolic versus diastolic pressure. That's because there's the blood pressure is still pulsatile there, but by the time we get to the arterioles and the capillaries, it's no longer pulsatile, it just blood flows smoothly, and it's just one single number. And then at the venous end, it's about 20 millimeters of mercury, and it just decreases as we get into larger and larger vessels. By the time blood returns back to the heart, the blood pressure there is nearly zero. So just FYI, this basically describes your blood pressure gradient. Okay, so that's our capillary exchange there. Now this is what bulk flow looks like, but capillary exchange in general, it refers to exchange of nutrients and waste and your respiratory gases. So I do want to say that lipid-soluble things can pass through the cell membrane, so they don't have to be necessarily part of that fluid. They can just, enter the cell and exit the cell because they're lipid-soluble, right? Water-soluble things can either be part of the bulk flow, or they can rely on facilitated diffusion using protein channels and such. They can also rely on some active transport like pumps, or vesicular transport like exocytosis, endocytosis, pinocytosis. So those are other possibilities as well, but most of substances move in this bulk flow fashion. So that's that. So capillary exchange is going to be the last thing that I have for you today. And next time we'll look at next time. it's not going to be live, but I'll create a video for the rest of the chapter. Start looking at those blood vessels. Okay, there's a lot of them that you do have to memorize. So I'm not going to be pointing them out to you because they're already pointed out for you. You just have to memorize them. So go to that Google Drive, open that blank, those blank figures, and either print them out or electronically annotate them, and practice, practice, practice. You have to actually write it down. Don't just look at it, don't stare at it. You've got to write these down. You got to label it yourself. And that's really the only way to learn it. Okay. So I'm gonna go ahead and stop recording now and then answer any questions if you have. If I find out to stop recording.